Hi, I'm Meredith at the VC Tutoring Center, and today I'm going to explain how to do a dihybrid cross. So, with a dihybrid cross, you're dealing with two traits instead of one. And in this case, I've chosen to do purple and white flowers and a round or wrinkled seed. Now, you always want to make sure that you define your alleles. So, I've already done it. I've given the capital P for a purple flower, lowercase p for white, capital R for a round seed, and a lowercase r for a wrinkled seed. The next thing you need to do is make sure you have the genotypes of your parents that are being crossed together. So this first parent is dominant for purple flowers and recessive for the wrinkled seed. The other parent is dominant, but a heterozygous purple flower and a round heterozygous seed. So next you need to make sure you figure out the gametes for each of your parents. And when you do a dihybrid cross, it's easiest to use the FOIL method, just like in math. So you'll take your first two letters in each trait and combine them together to get capital P, lowercase r. And then you're going to do it again for your outer letters. Here it's the same, capital P, lowercase r. Inner, again the same thing, capital P, lowercase r. And then outer, again it's the same thing, capital P, lowercase r. So I'm only going to have one gamete from this parent. Then you do the same thing for the next. You do your first, giving you capital P, capital R. You're going to do your outer, giving you capital P, lowercase r. You're going to do inner, giving you lowercase p, capital R. And then you're going to do last, giving us lowercase p, lowercase r. So now that I've defined all of my gametes, I can go ahead and fill in my Punnett square. So along the top, I've chosen to do the parent that has 4 gametes. So I'll put each one down. And then along the side, I'll just use the one gamete from the other parent. And now we're going to do what we've done before, bring down and across each letter from each trait. Keep your P's together, so this will be capital P, and keep your R's together. Capital R, lowercase r. And continue to do the same thing for the rest of your Punnett square. So I'll just do it quickly. And at this point, you've determined what all the possible offspring will be. And now you're going to take a look at, again, your phenotype, what the offspring actually looks like. And when I do a dihybrid cross, I like to write down the possibilities for your offspring. So here, you could potentially have purple flower, round seed. So I'll write down purple and round. And then I'll go through and cross off each offspring that has that phenotype. So our first one, we already know has purple and round. Second one, does not, so I'm going to skip it. The next individual is still purple and round. So I'll cross that one off and mark it down. And then our last individual is not purple or round. So I'll go to my next option. So this offspring is going to be purple and have a wrinkled seed. So I'm going to write that one down and count up how many offspring there are of that type. So again here, purple and wrinkled. And then the last option is also purple and wrinkled. So you count it up, 2 that are purple and round, 2 that are purple and wrinkled, giving you your phenotype ratio of 2 to 2. And then you can always make that smaller. 2 to 2 is the same thing as a 1 to 1 ratio.